Alright, hey there. In this one, we are going to look at the DESeq2 analysis and how we can use the DESeq2 onto the files that we created in our previous video and get some meaningful data out of it. Analyze the RNA-seq data and get the fold change, the p-value and all that good stuff. So before we start, we need to load in our libraries. So these are the three libraries that we're working with. So I'm going to select all of them and hit Command Enter, and this is going to load the libraries. And the next thing that we want to do is we want to set working directory as the same folder in which we have the files. So for me, it is already open. I'm just going to go to More and hit Set as Working Directory. And it's going to set this path as the working directory. Now we're good to go. So the very first thing that we need is to call in the counts data and the design file or the sample info. So to do that, I'm going to create a variable first. So counts underscore data. And inside this, I'm going to pass in the command of read.csv because we are reading the CSV file. And inside this, in quotes, we are going to write the name of the file. So this is counts underscore data.csv. And remember, you need to specify the file format in order to get the file inside your environment. I'm going to hit Command Enter, and you can see we have the counts data loaded onto our environment. I'm going to hit on this, and this is the counts data where you have the samples on top as rows, the genes as the samples as columns, genes as the rows. So these are ensemble gene IDs. You have the samples on top as columns, and you have the read counts of each of the genes in the samples. So the next thing that we want is the design info, which matches the counts data. So to do that, I'm going to say call data. So this is again this is a variable that I'm creating. You can name it anything. Call data is equal to read.csv once more because it is a CSV file again. Inside this, I'm going to say sample underscore info.csv. Go to the end, hit Command Enter, and you have the call data loaded. I'm going to open this up. And you have the different samples as rows over here. And you have the two columns as cell line and the dexamethasone, which comes in from the Airway package or the Airway dataset. So the next thing that we want is to check whether the row names of the call data, that is the row names or the sample names that are there. So this is these are the names of the rows. So are these matching with the column names of our counts data? These should be the same samples as that of the call data. We need to check that. And one more thing is the order of the samples that are present in the call data. So we have something ending with 08 and 09, 12 up till 21. So is this in the same order as that of the column names of this counts data? We need to check out this too. If it passes true, then we're good to go. All right. So first, we're going to check whether the row names in the call data matches the column names in the counts data. So as simple as that. So all call names inside the counts data, we need to write percentage in percentage. We're just checking whether it is there inside the row names of the call data. So inside row names of the call data, right? If this passes true, then we're good to go with this statement. So Command Enter, and you can see over here in the console it passes true. So now we are sure that all the column names in the counts data are there in the row, are there as row names in the call data. Now I'm going to check the order. Do that again. We're going to write something similar. So all call names in the counts data all call names in the counts data. To check whether they're inside, we need to hit 2 equals to. We're checking whether this is the same order as that of the other file. In order to do that, we need to hit 2 equal signs. So are they in the row names of the call data? So if this passes true, then again we are good to go. So command enter, and you can see it passes true. That means whatever row names over here is present in this one, and it is present in the same order. That is from top to bottom in this one, that is from left to right in this one. So now we can go on with the analysis. Okay, so the next thing that we want is to create a DESeq object. Okay, so to do that, we're gonna call in a command from the DESeq2 package. So DESeq data set from DESeq dataset from matrix, the second one. So we're gonna pass in some arguments. So we're gonna pass in the count data is equals to our count underscore data that we have over here, and next we're gonna say the call data is equal to the call data variable, and our design factor is dexamethasone. So we're gonna give a squiggly line, which is just above your tab key on the keyboard, the shift and the key just above your tab key. That is going to give a squiggly line, and we're going to say dexamethasone. So we need to be very careful of the spelling mistakes, otherwise it won't work. And I'm gonna put this whole thing, because we're going to use it again and again, inside a variable called DDS. And go to the end, command enter, and we have a DDS object created. This is the DESeq2 object. Okay, we're going to run some commands on this. You can see the DDS object. We are interested in the call data, and we are interested in the assays. Okay, so the next thing that we want to do is we want to filter out some rows with low gene counts. Before that, I'm going to show you the DDS object over here. There is a DESeq data set. It has got this many number of rows and 8 columns, and you have the call names as the sample names, and the call data names as cell line and dexamethasone, what we passed on from our design file and the counts data. Now what I want is to filter out or to remove rows with low gene counts. Essentially what I'm going to do is we'll keep all the rows that have at least 10 reads in total and above that, so not below that. Whatever is below 10, we're going to remove them. So I'm going to create a variable again, keep, and row sums in count of DDS, and I'm going to put a condition of greater than or equal to 10. If it is more than 10, keep them, otherwise remove them. So if I do this, you can see it's loaded as another object, and what I want you to see is it has got 63,677 as the number of rows right now. So if I hit DDS now after this, and I'm going to assign whatever I kept from this inside DDS and remove the others. So DDS square brackets keep and comma. So if I run this, and now if I hit DDS, you can see it has reduced to 22,000. Right, so we have filtered out a lot of rows with the gene counts which are lower than 10, with the read counts which are lower than 10. Okay, we're almost there. So now we have to set the factor level. Factor level is like the treated and the untreated, which is going to be analyzed on the basis of what. So our reference would be the untreated. We're going to see the comparisons with reference to the untreated, and what is the differentially expressed genes in the treated compared to that of the untreated. So to set the factor level, we are going to subset DDS, and we're going to subset this by dexamethasone. And we're going to set this to relevel DDS, again dexamethasone. We're going to set the reference equals to untreated. So this is going to set the reference as untreated. Okay, so now if we hit DDS, you're going to see the same thing, but what we're going to do is we're going to run the DESeq command on the DDS object that we created. So DESeq inside brackets DDS, and we're gonna reassign this value, reassign whatever we get inside DDS again. So we're gonna assign this to DDS and hit Command Enter. Okay, so these are the different steps that are required to, analyze the data that we just gave inside DDS, gave inside DESeq, and this is going to give us another table which we can view with results of DDS. If we hit Command Enter, then you can see over here, this is the result, and we have the p-value, the p-adjusted value, and the log2 fold change. So these are the things that we're interested in. So what is the fold change of a particular gene compared to that of the untreated? How much it has changed, whether it is upregulated or downregulated. We're going to know that from the fold change, and you're going to visually see this when we make the volcano plot. So now we have the results. I'm going to assign it to a variable called res. I'm going to hit Command Enter once more. So whatever you saw over here is now inside res, and we can hit a summary of the results. Command Enter, and you can see that the p-value is it has calculated the results by putting the p-value less than 0.1, which might be okay for you, but you can make it more stringent by making the p-value lesser. We're going to do that in a bit. So normally we'll put it as 0.05 or 0.01, not as 0.1. To change the p-value, we need to write results inside that DDS. I'm going to set the alpha equals 0.01 or 0.05, whatever suits you. So for this one, I'm going to write 0.01, and I'm going to hit Command Enter. But before that, I'm going to assign this to a variable called res 0.1, and I'm going to hit Command Enter. So now res 0.1 is having this. So again, if we hit summary of res 0.1, Command Enter, you can see the p-value, the adjusted p-value is 0.01. If it suits you, you can make it 0.05 or whatever you want. So these are the different parameters upon which you can check. So this is the upregulated, this is the downregulated, and these are the percentages of those. So the only thing that is remaining to visualize the data is to make a volcano plot or a MA plot, which we are going to do in the next video. So I'm going to see you there. Bye-bye.